Hi, everyone. So today we're going to talk about microbial nutrition, ecology, and growth. This is a continuation of Chapter 9. And today we're going to cover Part 2, environmental factors that influence microbes. So these are the learning outcomes for this section. By the end of this section, you will be able to um, list and define 5 terms used to express the microbe's optimal temperature growth or optimal growth temperature. Summarize 3 ways in which microorganisms function in the presence of oxygen. List other factors that affect microorganisms in relationship to their environment. List and describe 5 major types of microbial associations, and just describe the characteristics of biofilms. Biofilms are really important in human disease, and they also just occur a lot in nature as well. So it's just kind of an important thing for us to be aware of. So here are some of the environmental factors that can influence microorganisms. So heat and cold or temperature, of course, can affect the growth of microorganisms. Various gases, particularly oxygen and carbon dioxide. Acids, whether something has a high or low pH, so alkali or acidic conditions. Various types of radiation can affect the growth of microorganisms. Osmotic pressure, how much salts or solutes are present in the environment can affect their growth. Hydrostatic and other factors can all affect the growth of microorganisms. And of course, if you want to optimize their growth, you're going to need to make sure that you're in the right temperature range, provide the right nutrients, provide the right osmotic pressure. All of those things need to be taken into account in order to have the optimal growth. But we can also use this information to control the growth of microorganisms, and we'll see that later in the course when we get to Module 3 and we learn about how to control microorganisms using some of these environmental factors. So one of the big ones and the easiest one is temperature. So microorganisms cannot control their temperature. They don't have any capacity to be able to maintain their body, their cell temperature. So they will assume whatever temperature is in their microenvironment, in their habitat. And so generally, most organisms will have a range of temperatures that they are able to grow in. So everything can't grow at any old temperature. There's specific temperature limits that will enable things to grow. And we can divide these into their 3 cardinal temperatures. So microorganisms will have a minimum temperature that they can grow in, their optimal temperature, and a maximum temperature that they can grow in. And this is very dependent on individual organisms. So some organisms will have a wider range of growth, and others may have a very narrow range of temperatures that they can grow in. So you really need to consider what organisms you're talking about. So when you're looking at the minimum temperature of growth, this is going to be the lowest temperature that permits their continued growth and metabolism. Anything below that temperature and their activities will be limited. And when we think about minimum temperatures, this is really why we use a refrigerator, right? If we look at the microorganisms that mostly cause human disease, which fall into this mesophile range, this green line, this optimal range here, we can see if you get below about 10 degrees Celsius, they won't be able to grow anymore. Now, the thing about minimum temperatures cooling organisms down, that doesn't usually kill the bacteria, it just metabolically slows them way, way down so they can't grow. So we can exploit this to to maintain foods and things like that for longer periods of time because we can inhibit the growth of microorganisms at these lower temperatures. Um, of course, in their optimum temperature, this is where they're going to be the most happy and best able to reproduce and be the most metabolically active. If the temperatures rise and get too high and you exceed their maximum temperature, Then what will happen is the proteins, when enzymes and nucleic acids become denatured or inactivated and are no longer able to function, and the microorganisms will die. So usually if you get above their maximum temperature, that will actually kill microorganisms and prevent them from growing further. So just keep in mind that keeping things cold just slows them down but doesn't kill them, but often heating them up will actually kill them. And we can see in this graph, there's a number of different types of microorganisms that be classified based on their temperature requirements. So the first one here, the blue line, those are the psychrophiles. These are the organisms that really like cold temperatures. We have the psychotrophs. These are organisms that can tolerate cooler temperatures. Um, and are slightly cooler for their optimums than mesophiles. And then, of course, mesophiles are those that are most likely to affect humans because their optimum temperature just happens to be the same as our body temperature. Um, And then you have the thermophiles that can take a little bit of heat, and the extreme thermophiles that like it really, really hot, like boiling temperatures. Most things can't survive in these super extreme temperatures. However, microorganisms find a way. So the thermophiles and hyperthermophiles, these are the organisms that like it hot. So I have some like it hot, right? And so this is an image of a steaming, you can't really see the steam coming off of this pond in Yellowstone. And there are microorganisms that are growing in this um, very hot water. It's from uh, the thermal ducts that are there. And this is an electron micrograph of one of the organisms that grows there. This is an organism called Thermus aquaticus. And this is actually the bacteria that gives us an enzyme that we use in the polymerase chain reaction. It's a thermostable enzyme that's able to survive extreme hot temperatures. So this organism would fall into the extreme thermophile range. It's not just a thermophile, it's an extreme. It likes it really hot, near boiling temperatures. These kinds of organisms, again, aren't associated with human disease. They tend to live in water and soils near volcanoes, compost piles where it gets temperatures that will become very hot, and habitats that are exposed to direct sunlight or deep down in the oceans where we have these thermal vents and things like that that occur. So some like it hot, some like it cold. Those are the psychrophiles. Now notice it's not psycho, it's psychro. So you've got to pay attention to the spelling there. So psychro means organisms that like the cold. They will grow, these psychotrophs will actually prefer the cold and will grow in very cold temperatures, like in the snow where you can get what they call pink snow, and that's because of a pigment that's produced by some algae that can grow in the snow. We saw that this year in Colorado and parts of Arizona where they had an excessive snowfall last winter. And then in the late spring, because the air temperatures were warm enough and the snow was still there, these algae were able to grow out and you can turn it into pink snow, like you can see in this image here. Psychotrophs are those that can grow slowly in the cold, but they prefer to be a little bit warmer. So these things, though, can be of particular concern when we start to think about maintaining things in the refrigerator. So if you have some kind of a food that has a contaminant with a psychotroph, they can actually continue to grow in your refrigerator and will contaminate food and things like that. So some like it hot, some like it cold, but most things like it just right in the middle. And so these are just a sampling of a lot of different kinds of microorganisms, all of which prefer to be in sort of the middle range temperatures, and we call those mesophiles. This is the majority of medically significant microorganisms, so the things that we are concerned about as human beings that cause us disease or cause us harm potentially, that live in and on our bodies. their optimal growth is going to be between 20 and 40 degrees Celsius. And we can see there's some ranges of temperatures there as well. There's organisms that are thermoduric, and this is things like the Bacillus and Clostridium organisms that can form spores, as well as Giardia that can form a very stable cyst. And what happens here is if they're exposed to high temperatures for a short time, they can survive that and then go on to continue to grow and divide. So if you're not careful when you're pasteurizing food and things like that, if they're contaminated with these particular types of microorganisms, that won't be sufficient to destroy them. And we often will see this kind of contamination in certain food products. Other organisms that are like psychrotolerant, so these are organisms that can survive in the cold and they will continue to grow. And that's going to be things like Staphylococcus aureus as well as Listeria monocytogenes. These are two types of organisms that are frequently associated with food poisoning because they're able to survive the cold. So let's watch this short video on food pathogens and temperature, and hopefully this will give us a better idea of how this is happening.
Microorganisms reproduce by fission. Each parent cell increases in size and then divides to form two daughter cells. The progression is swift. 2 become 4, 4 become 8, 8 becomes 16, 16 become 32, 32 becomes 64, and so on. The growth of microorganisms depends on conditions in the environment. The main factors are temperature, moisture, nutrients, pH, oxygen, and chemical inhibitors. Each microorganism has very specific needs in each of these categories. The growth of microorganisms follows an exponential pattern. If there is sufficient water and nutrients and a warm enough temperature in the microorganism's environment, and the other three conditions are right, multiplication is extremely rapid. Under ideal conditions, the generation time for multiplication of bacteria can be as short as 30 minutes. So, in 24 hours, in the right environment, one bacterium can produce over 1 billion offspring. Of all the environmental factors that influence bacterial growth, environmental temperature is the most important. The following graphs depict the change in growth rates of typical microorganisms as a function of temperature. Here is a typical graph showing the increase in numbers of cells of the bacteria called E. coli as a function of time at the optimum growth temperature for the organism. Now watch the way that higher temperature affects the survival of E. coli. As the temperature is increased above the maximum growth temperature, the E. coli cells begin to die. As the temperature is increased, the cells die more and more rapidly until all cells are dead at temperatures above 80 degrees Celsius. Now watch the way that lower temperatures affect the growth of E. coli. As the temperature is decreased, the rate of growth slows until at some low temperature, slightly below 6 degrees Celsius, the cells cease to divide. The cells are still alive and waiting for the temperature to increase so they can begin to divide again. It is the cessation of growth of microorganisms at low temperature and their physical death at high temperature that defines the safe temperature ranges for the storage of foods.
So I think that that's something to really keep in mind is the fact that refrigeration and cooling of foods doesn't kill bacteria. It's actually a way to sort of preserve it. And this is why in your laboratory activities, you might set up a plate, make a street plate or something like that, and you put that in the incubator. The next day, your professor is pulling that out of the incubator and putting it into the cold so that when you come back the following lab, the bacteria is ready for you to work with and it's been slowed down in its growth. If it were left, those petri dishes were left at room temperature or in the incubator at the higher temperature for the whole week, they would be so overgrown. Most of the organisms will be dead because the agar will get all dried out. The bacteria will overgrow and use all the nutrients, and you just wouldn't really be able to have much of an experiment there. So that's why we put things in the refrigerator to cool things down.
Microorganisms.
So that's temperature control. The next one is gases. The two main gases that we consider are oxygen and carbon dioxide. And we're going to talk about oxygen first. So oxygen has the greatest impact. And you may not know this, but oxygen is actually can be highly toxic to microorganisms as well as to humans as well, for that matter, even though we need to have it. We can Microbes can fall into one of three categories: those that can use oxygen and detoxify it, those that can neither use oxygen nor detoxify it, and those that don't use oxygen but can detoxify it. So we'll talk about each of those groups. So how do microorganisms process oxygen? When it enters into cellular reactions, it gets transformed into multiple toxic products. And one of those is singlet oxygen, which is depicted as O or 1O2, as we see here. This is an extremely reactive molecule. It's like ready to bind and interact with other other molecules that are in the cells. And it is produced naturally by living things through our natural processes. And we actually use it, our phagocytic cells use it in some of uh, our systems in order to kill invading bacteria when they form the phagolysosome. Some of those packets will contain a singlet or reactive oxygen. If you get too much of this, it can damage and destroy a cell. Other ways that we can process oxygen, so if we have these superoxides and we have these toxic things being produced, either superoxides or hydrogen peroxide and hydroxyl free radicals, these are all destructive metabolic byproducts of oxygen and metabolic processes involving oxygen. Fortunately, many cells, including microorganisms, have enzymes that are able to scavenge and break down these toxic oxygen byproducts. And these are just depictions of the two enzymes. This is superoxide dismutase, and this is catalase. This is to remind us of the tertiary structures of proteins. You don't need to memorize these or really pay too much attention to them. I just wanted to point out, that these are what the structures of these proteins, that these enzymes look like. And so what happens is you have the first step is to use superoxide dismutase, or SOD, and it will take the oxygen-free radicals, the two O2 minuses, plus two protons, two H pluses, and turn that into hydrogen peroxide and O2. Of course, O2 is fine. And then that hydrogen peroxide will further get broken down using catalase to break that down into water and oxygen gas. We'll see in the laboratory that some bacteria can produce catalase, like Staphylococcus, and other bacteria cannot produce catalase. And so you'll actually do a catalase test in the laboratory to distinguish between different types of microorganisms. Now, many microorganisms can produce catalase, but some cannot. So we can categorize microorganisms based on their oxygen requirements. And so we've got a number of different levels here. The first are the aerobes. And so aerobes are those that will use oxygen and will carry out cellular respiration using oxygen as a final electron acceptor, and will function kind of the way that we think about our own cells functioning as far as oxygen goes. Some are obligate aerobes, and an example of that is Pseudomonas aeruginosa. And so if we look at these tubes over here, these are thioglycolate tubes, which contains a reducing agent that kind of pulls the oxygen out of the liquid media here. And we can see for an organism like Pseudomonas aeruginosa, it's going to grow just at the very top of this tube. It cannot penetrate or grow any lower because it has to be in an oxygen environment. The next two tubes represent facultative anaerobes. And so these are organisms that can function either in an oxygen environment or without oxygen. They're able to grow in either condition. They have the enzymes that they need in order to break down the oxygen toxic particles or toxic components, but they don't need to have oxygen in order to grow. And so here we have Staph aureus and Escherichia coli, and we can just see how these grow. Just kind of show different growth features in this thioglycolate media. And then we have strict or obligate anaerobes, and that's in this final tube where we can see Clostridium. And notice there's a space between where the thioglycolate media is and where the bacteria is growing to get to that really condition of no oxygen being present because oxygen is actually toxic to these organisms. They don't have the capacity to um, take care of those free radicals and superoxides and things like that that get produced through by oxygen. So they have to be sequestered and removed from the oxygen. And then there's others, microaerophiles that really just need very small amounts of oxygen. And then, of course, anaerobes, meaning without oxygen, so without oxygen. And then there's the aerotolerant anaerobes, those that can tolerate some oxygen, but they really prefer to be in a situation without it. So now that we've considered oxygen, we should also look at carbon dioxide. So this is just a CO2 molecule. 3 minus carbon plus 2 oxygens is CO2. Everybody needs some carbon dioxide in their metabolism. All microbes do. But some need higher levels of carbon dioxide in order to survive. And those are called capnophiles. And so they require higher CO2 tension. So for those, if you want to culture those in the laboratory most efficiently, you need to actually connect your incubator to a tank of carbon dioxide. And those are called CO2 incubators. And this is just a picture showing us that this incubator is being held at 37 degrees Celsius, and it's maintaining a CO2 concentration of 5%. Typically, if you're growing human cells, you grow them in the same condition, 37 plus 5% CO2. Now, in our laboratory, we don't really need to use the CO2 incubators because most of the things we grow don't require that higher CO2 tension. They're fine with just the amount of CO2 that's in the atmosphere. So some important pathogens that are associated, though, that are considered capnophiles that would require CO2 incubator to grow are Neisseria, Brucella, and Streptococcus pneumoniae. So another thing that we can consider in culturing microorganisms is pH. pH refers to the amount of hydrogen ions or hydroxide ions that are present in that media or in that condition. So it's a measure of acidity or alkalinity or how acidic or basic a solution is. And so when you're at pH 7, that would be pure water, H2O, where there are an equal number of hydrogen ions compared to hydronium ions or hydroxide ions. If you decrease the pH towards 0, you are increasing the amount of hydrogens that are present. So as pH decreases, the acidity increases because you're increasing the H plus that's in that solution, and that's going to make it more acidic. So if anything with a pH less than 7 is considered an acid, and anything with a pH greater than 7 is considered a base or basic. And the other word for that is alkaline or alkalinity. And so here you're increasing the OH in that compared to the hydrogen. So less H pluses and more OHs, and here it's more H pluses and less hydroxide ions. See the difference here. So most of the things that we're familiar with grow in sort of a neutral pH in the 6 to 8 range. There are some exceptions, like there's Helicobacter pylori that lives in our stomachs, or can live in our stomachs, and that can survive in a pH of 2, the acidic environment of our stomachs. but most things don't really like to be in that acidic of an environment. There are some extremophiles that like to live on the edges. This is microbiology, so there's always these things that like to live out in the edges, the extremes of the environment. And so one is the obligate acidophiles. These are organisms that require an acidic environment. Some examples of this are Euglena mutabilis and thermoplasma, an algal cell and an archaea. And they can tolerate, or they need to be in an acidic environment. A lot of molds and yeast can tolerate acid, and this is why they are common. This is what will spoil your pickled food. So if you have some kind of a pickled vegetable or something like that, and it becomes contaminated, it's most likely gets contaminated by a mold or a yeast. Most bacteria aren't as able to live in this extreme acid condition. On the other hand, there's some microorganisms that like to be in the alkali condition. They like to be in high pH. They live in the hot pools and soils where the environment is very basic. And an example of that is Nitronomonas species. And also the organisms that decompose urine create an alkali environment in order to balance the pH there. So another condition that we need to consider is osmotic pressure. Some organisms I put in there, where'd it go? Some like it salty. And this can be salty or sugary. So it's just having a high solute concentration. So osmophiles live in habitats with high solute. Generally in the environment, that's going to be salt. But we also can look at sugar because we increase osmotic pressure when we use high sugar. And that's really how we preserve foods if we make jellies and jams and those kinds of things where we're using high sugar to preserve food. So some of these can be obligate halophiles. If it's a halophile, halo is definitely salt loving. Osmophile can be salt or sugar, like just high solute concentration. So Halobacterium and Halococcus, those are both types of archaea. They like to grow in very high concentrations of salt, 9 to 25%. So the ocean is maybe 3 to 5%. This is like Dead Sea, uh, the Great Salt Lake, places like that that have very high salt concentrations. And they have modifications to their cell walls. They won't allow them to grow in hypotonic conditions. We also have facultative halophiles, and one is Staphylococcus aureus that is a human pathogen that we will hear a lot about. We've probably already stained it in the laboratory. These organisms can grow in high salt and are tolerant to salt, but they don't need to be in high salt. And if you think about where in and on our bodies we find Staph aureus, we can find them growing on places on our skin where we might have a lot of perspiration and things like that. And perspiration tends to have high salt concentrations. And in fact, that's one way we can distinguish Staphylococcus from Streptococcus is Staphylococcus can tolerate high salt, whereas Streptococcus cannot. So other considerations, radiation, hydrostatic pressure, and moisture. So organisms can tolerate, there's some organisms that can tolerate radiation and can survive in radio conditions where they're exposed to ultraviolet or ionizing radiation, gamma rays, that sort of thing, which that's an area of people who study those types of organisms are kind of thinking of those in terms of for astrobiology, like if these things can survive in that kind of an environment, might they survive out in space somewhere or out on some other planet or something like that? So that's something to kind of think about. We have the non-photosynthetic microbes that are damaged by toxic Oxygen can often produce products by contact with the light. So some of the yellow carotenoid pigments that we see will protect them from getting damaged by light and dismantling the toxic oxygen. Another thing to consider are barophiles. These are found that can tolerate high pressure, and you'll find these deep down in the ocean, where these extremophiles, usually archaea, are the most common that we see there that can survive in those high pressure conditions. All cells require water. Everybody has to have water. If we dehydrate cells, then that's often a way that we can kill them. And sometimes we can use them to store them. So, what are some of the ecological associations among microorganisms? We've talked about sort of what their environment needs to be and all the different factors that go into that. But in our classroom, we tend to look at organisms just by themselves, a plate of E. coli or a plate of Staph aureus or whatever. But in the world, microorganisms exist in shared habitats. They exist in relationship to others in some way. And so this table is showing us kind of what those different kinds of relationships can be, and we'll sort of talk about those as we go through this talk. So these interactions that these microorganisms can have can be beneficial, harmful, or neutral um, among the organisms that are involved. They can be obligatory or non-obligatory. So some associations you've got to have it in order to survive, and others aren't necessary. And sometimes they can involve nutritional interactions. So let's go through these. So the first one is the term symbiosis. Symbiosis is generally a term that's used to denote a situation of organisms living together in a close partnership. This can be a positive or a negative um, situation. So it's not always a positive. It can also be a negative for one or other of the organisms involved. So this is an example of a of a of a symbiotic relationship where we have this organism in these root nodules. So if we look at this is a plant that's been taken out of the soil, and here's their roots, and you can see these nodules here. These root nodules on these clover roots contain the bacteria in the genus Rhizobium. And this is a bacteria that is a nitrogen fixing organism. So a little while ago, when we were talking about nitrogen, and we said there are organisms that can pull nitrogen out of the atmosphere and convert it into a usable form, into an NH3 or NH4. This is the organism that can do that, and they live often in the roots of plants. And that is a mutualism where both organisms are benefiting. The bacteria is benefiting because the plant is providing it with all the other nutrients that it needs. And the plant is benefiting because the rhizobium is providing it with usable nitrogen so that it can grow. Another example of symbiosis is commensalism. And so what happens here in commensalism is the relationship doesn't necessarily benefit one member. It can benefit one member and generally not the other. So the commensal usually benefits or receives some kind of a benefit from the partner. The co-inhabitant maybe isn't harmed, but it also probably doesn't benefit. If it's a parasitic relationship, so parasitism, which is another type of symbiosis, this is a negative relationship. So here, the host is providing all of the nutrients or all of the things that the parasite needs, and the parasite is just taking, and it usually harms the host. So this is parasitism is an example of where symbiosis is a negative, has a negative relationship for one of the members of that relationship. In commensalism, and here's an image here, this is actually a test that is done to look for the bacteria Haemophilus. So Haemophilus influenzae will grow and form these satellite colonies around Staph aureus. So in the clinical lab, if you suspect a patient has a Haemophilus infection, maybe causing their respiratory disease, you could take that sample that you suspect is Haemophilus and culture it with you lay down a streak of known Staph aureus and then culture the Haemophilus around it. And if the Haemophilus grows, you'll see these satellite colonies that are growing. And this is because the Haemophilus can't grow on 5% blood agar, on the regular blood agar, but it can grow where around the Staph aureus, because the Staph aureus, when it breaks down the red blood cells, it produces NAD as a byproduct, and that can get used by the Haemophilus. So this is known as satellite growth or satellite colonies, and is actually a way to diagnose this organism, Haemophilus, and it's a condition of commensalism, an example of commensalism. So let's watch this short video. It's not so much about bacteria, but it does talk about all the topics that we've just mentioned, and we'll see how it goes.
We're in the ocean of Southeast Asia and getting ready to search for a fish that lives in a place so dangerous that most other fish won't even come near it. Ready? Let's go diving.
And after a bit of searching, I finally found it. The clownfish, or anemone fish as it's also called. The beautiful tentacles of the anemone are actually deadly to most fish. The tentacles have stinging organs called nematocysts, which fire tiny darts into fish that brush by. This stinging ability protects anemones from predators and help them capture prey for food. But how can clownfish hide in such a dangerous place?
Well, that's because clownfish have a symbiotic relationship with anemones that is so important that clownfish are almost always found among anemones. Symbiotic relationships are interactions between two species that can benefit either one or both species. Symbiotic relationships are divided up into three major categories.
Parasitism benefits one species while the other is harmed. Like ticks, for example. There are many examples here in the ocean. Take parasitic fish lice, for example, that cling to a host fish and feed on its body fluids. Amazingly, fish will sometimes find a cleaner fish or a cleaner shrimp to help remove the parasites from their scales, mouth and gills. When this happens, a mutualistic relationship is formed between the fish and the cleaner organism. Mutualism is a symbiotic relationship in which both species benefit from the relationship. This bird here eats the tick, which benefits the cow. In our fish example, the host fish is clean off its parasites, and the cleaner fish gets a free meal. Both species benefit, but not the sea lice anymore. Hey, I already brushed my teeth today.
The third symbiotic relationship is commensalism. Commensalism benefits one organism, but the other one is neither harmed nor helped.
You might have had a bug hitchhiking on you at some point without knowing it. An example here in the ocean is when remoras hitch a ride on sharks, big rays or turtles. The remora attaches itself with a disc on its head and then hitch a ride and feed off the food scraps that the host animal leave behind.
So the sharks are not affected by the remoras being there, but the remoras get a free ride and a free meal. Sounds good to me.
So back to our clownfish. Clownfish are protected from predators by living among anemones, and in exchange the clownfish chase away fish and other animals that could harm the anemone. What kind of symbiotic relationship is this? That's right, mutualistic. Okay, but we still have one important question to answer.
How can the clownfish be immune to the anemone sting? Well, a clownfish must first get used to its host anemone by developing a protective mucus coating, kind of like when we use sunscreen. And there are two possible explanations for how this happens. The first explanation is that the clownfish secretes its own mucus, which lacks the substance that makes the anemone fire its stinging nematocysts. It's as if the clownfish had a protective force field or invisible cape. And the second explanation is that the clownfish uses mucus produced by the anemone. The unprotected clownfish initially touches the anemone with its fins, even getting stung slightly at first, but over time gains the protective mucus coating. Symbiotic relationships happen everywhere, even in our own bodies. Did you know that every time you eat yogurt, you're actually filling up your digestive tract with symbiotic bacteria? That's right, you become the host for these natural bacteria, and they help you process and digest the food that you eat every day. Alright, so next time you go outside, stop and look around and see how many symbiotic relationships you can spot around you and record what they are. And remember, never stop exploring your world.
So even though that video doesn't really show us about bacteria per se, other than the very end where he talks about the yogurt, I think it's a really good demonstration of those three types of symbiotic relationships and the way he describes them. Plus, you get to see cute fish and things, so what can go wrong there? And microbiology is a type of biology, right? So we can also have non-symbiotic associations. And here we can have antagonistic or synergistic relationships. So in an antagonistic relationship, what happens is one member, the members are competing for nutrients and things like that. And one member will secrete a substance, often an antibiotic, that will prevent then the growth of that other organism. And you don't really need to worry about the details of this image. You can read about it if you want to in the notes page, on the notes part of the PowerPoint. I have posted that information there for you. But really what this is showing is, under these different types of plate conditions and the different mutants or whatever, basically the organisms are producing some kind of an antibiotic that's preventing them from being able to grow over each other and come in contact with each other. And so this is a type of antagonism. And when we think about antibiotics, and we learn about antibiotics later in the semester, that's why organisms are producing them. We'll see that most antibiotics are made by either bacteria or fungi. And the reason is to protect themselves in their environments and keep all the nutrients and keep all the stuff for themselves and prevent others from kind of taking over their little microenvironments. Synergism, on the other hand, is where organisms function in such a way that they all benefit, that they both benefit from the relationship. It's not necessary for their survival, but when they find each other, they get to a beneficial situation. And a very specific type of that is called a biofilm. And this is, the kind of the definition of synergy and how microorganisms can live together in a microenvironment. And sometimes that microenvironment is our teeth, or if we have some kind of an indwelling catheter or any kind of an implant or something, a biofilm can form on that. We see biofilms if you get kind of that slimy layer in your drain, in your sink, or on your shower curtain. That's a biofilm. They occur naturally in nature, on ponds and in the soils and things like that as well. So these are things that occur in a lot of different environments. And what happens, this is kind of the steps of how this occurs. The pioneer bacteria are the first ones to colonize the surface of whatever that surface might be. It could be the surface of your teeth, for example. Other microorganisms then will, the pioneers will start to secrete some extracellular materials, and they may have, slime layers or some kind of a glycocalyx that makes them sticky and adhesive, and they can secrete other attachment and adhesive qualities to them, and that will attract other microorganisms, and then other microorganisms will join, and then you end up with a mature biofilm where you have, you know, these organisms in the biofilm are going to be using quorum sensing. And we're going to watch a couple videos on quorum sensing because this is a really kind of an important concept. But what will happen is they will, you know, figure out how many organisms are there or what types of organisms are there. They can use quorum sensing for both of those things. Those are ways that microorganisms can use chemicals to sense how many other organisms like them are around, or how many organisms that aren't like them are around. And they can use these chemical signals to determine when to start releasing things. And that's, you know, kind of how they can end up causing diseases and things. Sometimes mature biofilms can stop quorum sensing, and they can start to send out new pioneers to start forming new biofilms. So I wanted to show you this video on quorum sensing. I know this is kind of a video-rich lecture this time. I hope you like the lectures or the little videos. I think they're useful. This is a short video from Bonnie Bassler. You can Google her if you're interested. She's got some really wonderful longer videos where she talks about quorum sensing and things. But I think this one really kind of puts it in a nutshell. It's a little bit long for us. It's like 7 minutes, but it's still, I think, worth watching.
So now what we understand is that all bacteria can talk to each other. They make chemical words, they recognize those words, and they turn on group behaviors that are only successful when all of the cells participate in unison. And so now we have a fancy name for this. We call it quorum sensing. They vote with these chemical votes, the vote gets counted, and then everybody responds to the vote. And what's important for today's talk is that we know that there are hundreds of behaviors that bacteria carry out in these collective fashions. But the one that's probably the most important to you is virulence. So it's not like a couple bacteria get in you and then they start secreting some toxins. You're enormous. That would have no effect on you. You're huge. But what they do, we now understand, is they get in you, they wait, they start growing, they count themselves with these little molecules, and they recognize when they have the right cell number that if all of the bacteria launch their virulence attack together, they're going to be successful at overcoming an enormous host. Hi, I'm delighted to be back to give you a little progress about what we've been doing in quorum sensing. And so today I want to tell you one story about how we're taking what we learned about these bacteria talking together and trying to interfere with that conversation to make a fundamentally new kind of antibiotic. And so the story I'll tell you about concerns this pathogen, Pseudomonas aeruginosa. This is the bacterium that kills people who have cystic fibrosis, it kills immune-compromised people, and it causes infections when you get a catheter, a stent, or a breathing tube. And the reason Pseudomonas is so virulent is because of this chemical communication, this quorum sensing. What Pseudomonas does is that as it grows, it makes and releases small molecules, which are the red triangles on this slide. And so as the cells grow, these molecules that are outside of the cells increase in proportion to cell number. And as you heard on the clip, when the bacteria detect that those molecules are there, they interpret that that means there's other cells around. And then as a collective, all of the bacteria together make a biofilm, which is how they sit on surfaces and adhere to tissue, and then the group together secretes the poisons, the toxins that make us sick. So that's quorum sensing. And so we want to be able to interfere with that conversation. And so we know what the molecule is that Pseudomonas talks with. It's the one that's on the left side of this slide. And so what we did using chemistry is we changed the structure of that molecule to make the one that's on the right. And so what that chemistry did was it changed the signal molecule the word, into an inhibitor. So we change the molecule that turns on quorum sensing into a molecule that shuts down quorum sensing. So what happens if you have such a molecule? So first I'll talk about biofilms. So in this Petri plate, what we've done is we've put Pseudomonas in the middle of the Petri plate. And what I hope you can see is that the bacteria have spread out to the edges. That's this biofilm formation. As a group, they move out over the plate, and that could be like your tissues. But we have this inhibitor. So now if we do the experiment, we put the Pseudomonas in the plate and we add the inhibitor, what you can see is that the Pseudomonas can't move. So that's good. That's step one in the infection. It seems like our inhibitor can shut down biofilm formation. The next question for us is, what about these poisons, these toxins that Pseudomonas secretes? So now you're looking at an experiment, and in the left-hand test tube, that's wild-type Pseudomonas. It's doing quorum sensing, and it's secreted these toxins. And when it secretes those toxins, the bacteria turn green. In the middle test tube, that's a mutant that we've made, where we've knocked out its quorum sensing system. So that mutant has no communication, and what you can see is that the bacteria are colorless. They can't secrete the toxin, so they don't turn green. The right-hand test tube shows you wild-type Pseudomonas that we've added our inhibitor. And what I hope you can see is that the inhibitor greatly decreases the ability of Pseudomonas to secrete that green poison. So now we're in business. It looks like, at least in the lab, we can shut down biofilms and we can shut down toxin secretion. So what about in an infection? So in this experiment, you're looking at an animal model system that we have for Pseudomonas infection in the lab. And all we do is measure whether the animals are alive or dead. And so on the line that you're looking at, obviously, if we don't add Pseudomonas, the animals are perfectly fine. If we give a Pseudomonas infection, now what you can see is that all of the animals die within the first day after the infection starts. But if we do that, we give the Pseudomonas infection and we give that inhibitor molecule that I showed you, what you can see with the third line is that we can greatly improve the outcome for the animal. So in fact, we think now that there must be merit to this idea of interfering with chemical communication, and that maybe this could form the foundation of a new type of therapeutic. And so what we're doing in the lab right now is we're taking the molecule that I showed you, and we have to make it more medicine-like. We have to build in potency, and we have to make that molecule safe. The second thing is that we got inspired by that biofilm experiment that I showed you, and we're working with engineers now to try to embed those inhibitor molecules into materials. And the idea is that maybe we could make infection-resistant catheters or stents or breathing tubes. And then finally, I'm just telling you one little vignette that's about Pseudomonas. We work on lots of globally important pathogens in my lab, and we're having similar success doing these kinds of strategies in other bacteria as well. And then to finish, I just want to show you the two students who did the work, Colleen O'Loughlin and Knut Drescher. They both work in the lab, and I'm lucky to get to work with them every day. Thanks for having me back.
Okay, so if you're interested in this topic, you should definitely Google Bonnie Bassler. She has some really excellent, like hour-long TED Talks and things like that. Very dynamic speaker that really explains about quorum sensing. She even has one about how the quorum sensing works in bioluminescent organisms as well in the ocean outside of Hawaii and stuff. So definitely something of interest and relevant to our class, because developing new antibiotics and just a completely new approach to trying to develop an antimicrobial therapy is really important, as we'll learn later in the semester. So I just wanted to include this slide I have in the notes. On the notes page, it gives you all the information about it, but this is just giving us, if we look at a mature biofilm, they've got quorum sensing going on. There's extracellular DNA because some of the cells have died and they've released their DNA. So they're sharing genetic information through horizontal gene transfer. They've got extracellular matrices being produced. They've got reactive oxygen species being in their microenvironment. Some of these cells are tolerant, some are persistent, and just, this whole gamut of a mixed population of organisms. And you can imagine if we were to treat this with a normal, typical type of an antibiotic therapy, some of these organisms that are kind of metabolically inactive because they're down in the middle of this biofilm, they're not going to be affected by antibiotics because they're not going to be taking them in and be bothered by them. And that's part of why these biofilms are so difficult to treat. So that brings us to the end of this topic. Again, you should be able to answer these questions by now. So if you can't, go back through and see if you can find the answers. Also, remember, I do have some notes embedded in the PowerPoint. So when you're looking at my videos, please have the PowerPoint open as well. I noticed for Chapter 9 PowerPoint, I didn't put the parts in as well because the big Chapter 9 PowerPoint, probably because the videos I've embedded, is just way too big for Google to handle and won't let you open it directly. You'll have to download it and open it, or you can open the individual the shorter PowerPoints that are in our shared drive. All right, that's it for now, and I will see you next time for part 3 of this topic.